All right. Sorry I'm late. My meeting ran a little bit long. So, I'm going to continue. So we're going to continue from last time. Last time I talked about the different enzymes. Involved in prokaryotic DNA replication. Very quick review of those enzymes so you can orient yourselves with what goes where. Is at the very front here, you have helicase. Helicase's job is to unwind DNA, is to open up DNA. When you unwind DNA, again, this is a review of last time. When you unwind DNA, you create supercoiling that needs to be fixed. We have enzymes to do that. The one example they give here is DNA gyrase. However, there's a broader category of enzymes called what? That fix supercoiling. Topoisomerases, good. These little green things, SSB, those are your single-stranded binding proteins. These proteins, their job is to keep DNA single-stranded after it has been opened up so that you can replicate it. So those single-strand binding proteins keep it open, keep the DNA open so that you can replicate them. Let's see, this little thing here is a primer. Do they have primase here? Yeah, I guess it's right here. So in order to do DNA replication, every time you do DNA replication, you need a short RNA sequence called a primer in order to start DNA replication. That short RNA sequence is always located at the front of whatever you are replicating, and the enzyme that puts the primer there is called primase. So this enzyme here, primase, creates a primer so that your polymerase enzymes know where to do DNA replication. The main enzyme in prokaryotic DNA replication is DNA polymerase Roman numeral 3. It doesn't say it here, but it's DNA polymerase Roman numeral 3. That's the one that does the majority of your prokaryotic DNA replication. The reason why it's able to do most DNA replication is because DNA polymerase 3 has this special subunit called the beta sliding clamp. This is a subunit that only DNA polymerase 3 has that gives it what's called enormous processivity, meaning all this clamp does is it makes sure DNA polymerase 3 is stuck to the DNA template. It cannot fall off, and if it can't fall off, it's going to keep replicating and replicating. So the reason why DNA polymerase 3 has such enormous processivity is because of the beta sliding clamp. Last time, near the end, we talked about two other enzymes, talked about here, called DNA polymerase 1 and ligase. These enzymes only work in a process called Nick translation, which is a process of finishing off DNA replication on the lagging strand. The way Nick translation works is in regular DNA replication, you created Okazaki fragments on the lagging strand. So Nick translation's job is to fill in these gaps between Okazaki fragments. This is done first by the enzyme DNA polymerase Roman numeral 1. Again, this is all a review of last time, so I'm going a bit fast. DNA polymerase Roman numeral 1, its job is to fill in the gaps between Okazaki fragments and degrade any primers that it finds. Why do we need to degrade the primers? Because the primers are made of RNA, not DNA, so those have to be removed. So DNA polymerase 1 will fill in the gaps between Okazaki fragments, degrade any primers that it runs into, and fill in DNA in those spots where the primers used to be. Finally, after DNA polymerase 1 fills in all these gaps, DNA ligase seals the last sugar phosphate bond between Okazaki fragments together. That is ligating the Okazaki fragments together. After that, you have a closed nick, and therefore your your lagging strand is fully replicated. So we talked about that. We talked about the specific activities that DNA polymerase 3 has and DNA polymerase 1 has. Let me see, as an example here. We talked about how DNA polymerase 3 is made of several subunits. Each one has a certain activity, either a polymerase activity or an exonuclease activity. I'll talk about it more today when we talk about the Klenow fragment. But if you want to review DNA polymerase 3 and DNA polymerase 1, refer to your notes from the last SI session. All right, so I think that is everything we did last time. Let me make sure. Where's the Klenow fragment stuff? Here we go. Okay, Klenow fragment. So there's a special enzyme that exists called a Klenow fragment, but it is not a naturally occurring enzyme. What this thing is used for is in the lab, if we want to synthesize DNA on our own, like let's say we have a single-stranded piece of DNA that we want to replicate. But how do we do that on our own in order to study like enzymatic activity and things like that? Well, we could do PCR, and that's most of the time what we do. But sometimes, and especially a long time ago, we use something called the large Klenow fragment to do this. What this is it's a modified form of DNA polymerase 1. Okay, so the Klenow fragment is DNA polymerase 1, but modified. All right, well, we'll see how it's modified in a bit. When I jump down to DNA, or when I go to DNA polymerase 1, does it have the activities listed here? Of course it doesn't. Yeah, here we go. I said last time that DNA polymerase 1 has 3 activities on it. A 5' to 3' polymerase, which that activity is another way of saying it replicates DNA. It has a 3' to 5' exonuclease activity. That is your editing function. So sometimes the DNA polymerase makes a mistake. When it puts in the wrong nucleotide at the wrong spot, it uses its 3' to 5' exonuclease to remove the incorrect base. And DNA polymerase 1 has this third activity called 5' to 3' exonuclease. This is unique to DNA polymerase 1. This activity was used to remove those primers on the lagging strand. That is what 5' to 3' exonuclease activity does, is to remove primers on the lagging strand. So, DNA polymerase 1 has these 3 activities. If I want to replicate DNA in a lab, I only need the first 2 activities. I don't want that last one, because if I have that last activity on an enzyme that is replicating DNA in the lab, it's going to destroy all the DNA that it just made. Okay? Again, this is in the lab. It is different from a biological condition. So what they figured out what they could do is they could just take DNA polymerase 1 and cut off that part. Notice there's a big gap here. What they did is they cut off the part of DNA polymerase 1 that normally would do the 5' to 3' exonuclease activity, that would normally do the primer removal thing. Okay, so this thing on the left here is your large Klenow fragment. It is a special modified form of DNA polymerase 1 that can replicate DNA. And it does so because it still has the 5' to 3' polymerase activity, and it still has the 3' to 5' exonuclease activity, the repair activity, the editing activity. And it lost the 5' to 3' exonuclease activity because we don't want that if we want to replicate DNA in the lab. How did it lose that? Well, we can use trypsin to just cut that component off of DNA polymerase 1 to create the large Klenow fragment. So, what do you think the small Klenow fragment is? Probably the thing that was cut off. The small Klenow fragment is not shown here. It's the one that was, the small Klenow fragment is the one that has the 5' to 3' exonuclease activity. So the large Klenow fragment is DNA polymerase 1 with just the first 2 activities still on it. You might wonder, well, why not just use DNA polymerase 3, right? Why not just, if I want to do DNA replication, why not just use if I want to do DNA replication in the lab, why not just use DNA polymerase 3? Because it has the first 2 activities. Why do I bother using DNA polymerase 1? The main issue is the fact that DNA polymerase 3 is made of, 4 different subunits, which is very hard to actually keep together in an artificial situation. DNA polymerase 1 is different, right? How many subunits is DNA polymerase 1 made of? DNA polymerase 1, confusingly, is made of just 1 subunit, right? DNA polymerase 3 is made of multiple subunits. So DNA polymerase 1, because it is 1 subunit with 3 activities, We actually prefer to use DNA polymerase 1 in the lab, but as you saw what I described earlier, we have to modify DNA polymerase 1 a little bit because it has that annoying 5' exonuclease activity. We don't want that in our artificial enzyme, so we cut it off and we created a large Klenow fragment to replicate DNA for us. Okay, so that is what you see in artificial DNA replication. When we're not talking about PCR, this is what we're doing. Let's see if there's anything else. That is it for the large Klenow fragment. So, any questions on that? All right. Okay. Moving on. We talked about DNA polymerase 3 and 1. Yeah, we already mentioned a lot of these features of DNA replication. We mentioned that it was bidirectional, so I won't go over that anymore. A lot of these we talked about, so I'm going to skip it all. Yep, we talked about all these as well. That's a picture. Okay, some other features specifically for prokaryotic DNA replication. All those features previously, some of them are seen in eukaryotic, some of them are seen in prokaryotic. I would memorize everything that you had learned up to this point as specifically just being prokaryotic DNA replication unless stated otherwise later. Okay, so everything is still prokaryotic. This slide gives you a couple extra things with prokaryotic DNA replication. So let me get ready to draw something. Okay. Unless there's a picture here. There is no picture. 1 second. Okay. Let's see. Okay, so here's prokaryotic DNA. I know it's prokaryotic DNA because it's circular. Here is one origin of replication, or an OREC site. We're zoomed out a lot. This one origin of replication will have 2 replication forks that will replicate DNA in a clockwise and a counterclockwise fashion. However, because prokaryotic DNA is circular, there's a unique thing that happens with prokaryotic DNA during replication. In that, your 2 replication forks will eventually crash into each other if they go around the circle, right? It turns out they reach a spot opposite to the origin of replication called the termination locus, the termination spot, the TER locus, TER spot. It all means the same thing. All right, it's a specific set of bases that you see on the other end, always on the opposite end of the origin of replication in prokaryotic DNA. Your 2 replication forks are going to meet at this spot here, at this termination locus. The reason, another important fact about this termination locus is there's a special protein that likes to bind to this termination locus called the TUS protein. I don't know what the full name of it is, I just know it's called the TUS protein. And what this protein does, the TUS protein, and the TUS protein binds to the termination locus. What it has a special function called contrahelicase. Using, context clues and everything, it sounds like this protein's job is to counter helicase, right? What enzyme is at the very front of every single one of these replication forks? One of the enzymes is helicase. So if you want to stop DNA replication, when the replication forks meet together at the other end of the origin of replication, they will run into this protein called the TUS protein. Because this TUS protein is a contrahelicase, it will stop the 2 helicases at each of the replication forks, and thus you've stopped DNA replication. Okay? So prokaryotic DNA replication is stopped via this mechanism here. There's a termination locus, which is a set of bases on the opposite end of the origin of replication. And there's a special protein that likes to bind there to stop the replication forks by countering the helicases. All right, this is only for prokaryotes. Eukaryotes, they do not have this. All right. And again, a big piece of advice I usually give people when they get to chapter 28, 29, and 30 is you should be making different pages, should have different pieces of information, meaning you should have a sheet of paper by the end of this when you're reviewing. You should have a sheet of paper that only includes information on prokaryotic DNA replication. You should have a separate sheet of paper that only includes information on eukaryotic DNA replication. It's a simple thing, but I can't tell you how important it is to do this, because if you are either scrolling through the PowerPoints, you have to go back and forth from prokaryote to eukaryote, and sometimes they mix things together. So you have to flip between chapter 28, 29, and 30. But if you do it like this, similar sounding terms will be organized in the exact spots that they're supposed to be, and you can directly compare them to each other. So I highly stress that chapter 28, 29, and 30, you are organizing your what you learn into whether it's prokaryotic replication, eukaryotic replication, prokaryotic transcription, eukaryotic transcription, and then eventually prokaryotic translation, eukaryotic translation. I won't be doing that for you up here because I have to go through this linearly, but I hope you are doing this on your own. Okay, back to PowerPoint. Let's see. Now we get into eukaryotic DNA replication. You will find that the information you need for eukaryotic DNA replication is a lot shorter. Really, the only things you need to know are the different polymerases that you see in eukaryotes. And at the very end, you'll learn about telomerase. After those two things, then you basically have eukaryotic DNA replication down. So, to break it down, eukaryotic DNA replication is carried out not by DNA polymerase 1 nor DNA polymerase 3. Whenever you see the phrase DNA polymerase with a Roman numeral attached to it, you are talking about prokaryotic DNA replication. Whenever you see DNA polymerase with a Greek word or Greek letter attached to it, that is eukaryotic DNA replication. So, I hear that there's some new information this semester, so I don't know this new information exactly. I'm going to give to you what I always learned about this stuff, and then pay attention in class for any slight adjustments he might give you. Basically, for this section of the notes, I just memorized DNA polymerase alpha. Here's the facts about it. Okay? DNA polymerase alpha is unique because it makes its own primers. Contrast that with prokaryotes, where you needed a separate enzyme to do that. The enzyme primase was the one that made the primers in prokaryotes. In eukaryotes, some polymerases can make their own primers. DNA polymerase beta is similar to alpha, except that it is also involved in repairing damaged DNA. Blah, blah. Yeah, you don't need to know this. It used to say skip on here, but you don't need to know this. Okay. More polymerases. DNA polymerase gamma. This one is the one responsible for replicating DNA in mitochondria, and I think it might also do chloroplasts. But the key thing is that remember in eukaryotes you have organelles. You have the nucleus, you have mitochondria, and plants have chloroplasts. And mitochondria and chloroplasts have their own DNA that need to be replicated as well. So there's a separate polymerase that's responsible for specifically replicating mitochondrial and chloroplast DNA that is called DNA polymerase gamma. Now what I've learned historically is that DNA polymerase delta replicates the majority of the DNA in eukaryotes. I note, someone brought up to me, and I noticed it says here that there's some new information on here. Let's see. Apparently, and there's a scientific paper link attached to here, it's only responsible for replicating lagging strand DNA. I believe that this means that you have to memorize that fact about DNA polymerase delta this semester. So memorize what you have here. It won't be more complicated than what is listed on here. It'll never be more complicated regarding the eukaryotic polymerases than what is listed on here. So new to me, so new to you guys too, I suppose, DNA polymerase delta is responsible for only replicating the lagging strand. Another fact about DNA polymerase delta that is familiar to me is it has a special extra subunit called PCNA. I don't remember, oh there it is, there's the acronym for it, proliferating cell nuclear antigen. This is analogous to the beta sliding clamp that you see in prokaryotes, which you might remember that the beta sliding clamp's job in prokaryotes is to keep DNA polymerase 3 attached to the template DNA so it can replicate a lot of DNA. Guess what? pCNA does the same thing to DNA polymerase delta. It keeps DNA polymerase delta attached to DNA so it can replicate it. Again, I have no idea how it only replicates the lagging strand. You don't need to know how it only replicates the lagging strand, just the fact that it does. This second enzyme, DNA polymerase epsilon, I used to learn that actually we weren't really sure what it did, that it only replicated some DNA. This semester, brand new, apparently it only does the leading strand of DNA and is also highly processive. So it turns out that in eukaryotes, DNA polymerase delta and DNA polymerase epsilon share the work. Delta does the lagging strand, epsilon does the leading strand. You're good if you know that. I believe this is some pictures. Yeah, this is just some pictures of eukaryotic DNA replication showing Okazaki fragments, pCNA, where polymerase delta would go and polymerase epsilon would go. You can take a look at it if you want, but you're not going to be given any pictures like this and asked to point out which one is which on here. Okay. More stuff about Okazaki fragments. Again, this is all new information to me. If this shows up on the exam, I will review it during the exam review. But as you can see, it is not well known, the mechanism of how this works. So feel free to be entertained via the scientific information that Dr. Jesus will give you in class. That is not my job. So I'm going to jump down to the last bit of DNA replication with eukaryotes that's important. Yes? When is exam 3? Yeah, April 15th is what it says on the syllabus, so it is April 15th. The syllabus is always, it seems like a lot of people are confused. The syllabus dates, the ones on there, you can go check on Canvas. The syllabus dates for exams are the true dates and times that you have your exams. I think probably what's on the grade book is probably just old information, so ignore the exam dates on the grade book. The exam dates on the syllabus are the exam dates that you need to know. So April 15th. Other questions? All right, so, all right, do we have time for this? Yes. So, to finish up DNA replication in eukaryotes, there's a unique problem with eukaryotic DNA replication that prokaryotic DNA replication does not have. Prokaryotic DNA is circular. You can replicate it 1,000,000 times over and there'll usually not be any issues. Eukaryotic DNA replication, we have a problem. Eukaryotic DNA is linear instead of circular. So, what happens, and forgive me if he gives you an animation of this instead, I usually just draw this. So, 5 prime, 3 prime, let's see, template, prime, leading strand, lagging strand, let's see if I draw this right. Nope, that's wrong. Okay, so in eukaryotic DNA, because it is linear, you know, DNA replication still happens normally. There's a leading strand and a lagging strand. But what happens when you reach the very end of a linear piece of DNA in eukaryotes? Well, there's no problem on the leading strand. You can finish replicating it. However, on the lagging strand, remember that on the lagging strand, the way this works is that you have to replicate it in the backwards direction, meaning in the direction opposite to the replication fork. That's why you create Okazaki fragments, is because the direction at which DNA replication occurs on the lagging strand is opposite to the direction the replication fork moves in. Because of that, there always needs to be a primer put ahead of these Okazaki fragments. A primer would be here, another primer would be here, another primer would be here. Well, let me illustrate this differently. But this last bit here can't be replicated on the lagging strand in eukaryotes. Why? Well, there's nowhere to put a primer on the lagging strand in a linear piece of DNA at the very end of it, because this is empty. There's nowhere to stick a primer at the very end of a linear piece of DNA. If you can't put a primer here, a DNA polymerase doesn't know that it needs to finish DNA replication of this template strand at the end here. This region, by the way, this region of DNA at the very end of eukaryotic DNA is called a telomere. And this unique feature is only in eukaryotes because it's linear. So this fact that you cannot finish replicating this piece of DNA, what will happen? What will happen is that in the daughter cells, their genome, their DNA sequence will be slightly shorter. It will not have this part of the sequence replicated. It'll be slightly shorter by a couple tens of bases, all right? Which might not seem like much, and usually that isn't a big deal. Genes that, there aren't any important genes that are found at the telomeres. The telomere part of your genome sequence is usually just long repeating sequences that don't really mean anything. But every time that cell replicates, the next generation of daughter cells, their genome will be slightly shorter and shorter and shorter. It turns out that after, I believe, 30 to 50 replications, your DNA would have shortened so much that it would have lost the ability to replicate anymore. Or if it does replicate, a lot of mutations are going to happen. A lot of cancerous mutations are going to happen. Bottom line is, most cells will degrade. Their telomeres will degrade every time they replicate. And it'll probably be about 30 to 50 replications before it's so short that something wrong happens. Okay? That amount of time it takes, that number of replications it takes, to result in something wrong happening, such as a halt of replication, is called the Hayflick limit. That is the number of times a particular cell can replicate before its telomeres are shortened so much that it lost the ability to replicate again, or a bunch of mutations start happening. Okay, that is the Hayflick limit. And again, this only happens in eukaryotes. Now, this is not a problem in every cell in the body. Some cells, especially ones that are supposed to replicate often, have a special enzyme called telomerase. Cells that have active telomerase are ones that replicate a lot. Your germline cells, your immune system cells, your stem cells especially, those need to replicate a lot. Those are all good. And then some cells that are bad that replicate a lot also have telomerase, such as cancer cells can sometimes have telomerase, active telomerase. Now, what does telomerase do? Well, let's look at this. Here's our lagging strand. The top strand is our template, and the bottom one is our new DNA that is being made. Normally, it is unable to replicate this last part of the template strand. However, if a cell has active telomerase, telomerase just extends the template strand. Not with anything important, just a bunch of random RNA bases that don't matter. It extends the template strand. Again, we don't care what bases are added because the telomere usually doesn't matter anyway. So when it extends the template strand, now look at this, all this open space that primers and DNA polymerases can now bind to and finish replicating the lagging strand. So notice now you are able to extend your new DNA strand long enough that you covered the original length of the template strand, okay, before the telomerase extended it. So that's all this enzyme's job is. All telomerase's job is to prevent the degradation of telomeres by extending the template strand so that you can finish replicating it. The enzyme in eukaryotes that is responsible for finishing the replication is specifically DNA polymerase alpha. So make sure you know that as well. Telomerase, all telomerase does is it extends the template strand, but DNA polymerase alpha actually finishes replicating it. All right? So you learn about telomeres, you learned why telomere shortening is a bad thing and why it happens, and you learn about the enzymes that fix that. So, the specific, more information about telomerase, and you do need to know this information about telomerase, is that it is a reverse transcriptase. So I believe that might have been talked about later in detail. But bottom line, what a reverse transcriptase does is it uses RNA as a template to make DNA. So I'll go over that in more detail when we run into that. What else is important here? Yeah, that's about it. Oh yeah, and what cells you're going to find active telomerase in. The bottom line is, if it's cells that replicate very quickly, you need to have telomerase in there. Question?
Yeah, so the telomere is the shortest strand.
The telomere in this picture here is just, by definition, the word just means the very end of DNA.
Yes.
The template strand of the telomere is this one up here.
Yes.
And this one's the new strand of DNA that you're trying to replicate it. So the shorter one is the new one that you're trying to make.
So the telomere in general is just the whole...
It's just the very end. Yeah, the whole thing at the very end. Yeah. Question?
So is a new primer put on after telomerase extended?
Yes, exactly. Now that there's enough space for it. Yep, now that there's enough space for it, DNA polymerase alpha comes in, attaches a primer on the newly extended template, and now you can finish replicating the lagging strand. Yes?
So it adds a primer to the telomere.
Yes, exactly. Well, the telomerase just extends the telomere. DNA polymerase alpha then adds a primer to the newly extended part and then finishes replicating it. Yes. I believe, and DNA polymerase alpha, if you scroll back up there, you will find it has its own primase activity. Well, look at that. It's using its own primase activity to finish replicating the lagging strand. All right. Other questions? Yes.
Whenever he talks about the term recess, so recess is the original shorter.
Okay, yeah, recessed versus, I think, overhanging. So, in the future, if you ever see the phrase recessed strand or overhanging strand, don't overthink it. All recessed means is the shorter one. It is shorter by whatever reason. Shorter because of this, shorter because something was cut off. It doesn't matter. Whenever you see the phrase recessed, it just means the shorter strand of DNA during replication or in some diagram. Overhanging, therefore, means the overhanging part, the one that is extended that has not been replicated yet or transcribed yet. It doesn't matter. But the phrase recessed and overhanging just is relative length. Okay? I believe an old exam question, or it might have even just been last semester, is, yeah, you know what? Let me try to make one up here. Let me go to DNA polymerase 3 real quick. DNA polymerase 3. You see that it has an activity called 3' exonuclease, right? That activity's job is to remove incorrectly paired bases. So during DNA replication, the wrong base is added sometimes, and 3' exonuclease's job is to remove it. When you remove it, what do you leave behind? You leave behind the previous nucleotide with what functional group at its very end? A 3' OH, right? Whenever you remove a nucleotide, the previous one must have its 3' OH so that you can add a new nucleotide to it. That 3' OH, is that going to be on the overhanging strand of DNA or on the recessed strand of DNA during DNA replication? It'll be on the recessed strand because that's the one that is being replicated. So if you have a mistake on the replicating strand, you need to make it shorter. So in order for DNA replication to continue, new DNA is always added on the 3' OH of the recessed strand, on the 3' OH of the shorter DNA strand is all that means. Okay, so, eukaryotes, oh, well, look at that, there's a video. That probably is a better video than anything I could draw, so you could take a look at that. PCR, I believe I've talked about already, so I will skip that. RNA synthesis. Is this reverse transcriptase yet? Here we go, reverse transcriptase. Looking for one nice slide that summarizes it for you, otherwise I have to write it all down. Here we go, and I'll go back to the other slides in a bit. Okay, so there's an enzyme that exists called reverse transcriptase. Remember the central dogma? The central dogma is DNA is replicated, DNA can be transcribed into mRNA, and mRNA can be translated into protein. There is an enzyme that kind of goes backwards in the central dogma, and the enzyme is reverse transcriptase. Its job is to take any RNA and reverse transcribe it back into DNA. All right? A lot of bacteria has this, mostly as a way to create bacteriophages and as a defense mechanism. There's a million different reasons why it has reverse transcriptase, of which you don't need to know why. But what this thing does, biochemically speaking, biologically speaking, is it has these three activities. So let's break it down. Activity 1. Reverse transcriptase has the activity RNA-directed DNA polymerase. All this means is there is an RNA template and it creates DNA out of it, right? RNA-directed DNA polymerase makes DNA from RNA. That's the first activity of reverse transcriptase. The second activity is RNA's hydrolase activity. That's what the H stands for. What this activity does is after the first one takes an RNA template and creates DNA out of it, you have created an RNA-DNA hybrid bound to each other, right? Because it was reading the RNA template and attached DNA nucleotides to it. So you create an RNA-DNA hybrid. The second activity's job is to break that hybrid into two pieces. So RNA's hydrolase activity, its job is to remove the RNA from the RNA-DNA hybrid. The very last activity is now that you've created your brand new DNA that was made from RNA, this last activity, DNA-directed DNA polymerase, will look at that new DNA that you made and use that as a template to make more DNA. That is what the third activity of reverse transcriptase does, DNA-directed DNA polymerase. In fact, at that point, not only will that activity replicate that DNA strand, but the particular cell's own DNA polymerases will replicate that DNA strand as well. This one just helps that along. All right? So these three activities are very important to reverse transcriptase. These three activities are most commonly asked about on the exam in regards to reverse transcriptase, so make sure you know these. Let's see. Although this is true, be very careful. You do not need to know the reverse transcriptase mechanism. You do need to know this fact. It is annoying that it's on this slide, but this fact has shown up as an exam answer or an exam question. The fact that reverse transcriptase actually uses the host's own tRNA as a primer to perform reverse transcription. It's a small little fact. You do need to know it. But this whole pictures of mechanisms for reverse transcriptase, don't worry about it, okay, for the exam. Let me see if there's any other little things like that that is stated here. All right, I'll go over some more next time. So that's it for now.